project here at Tech that has as an objective to build a biobank of 100,000 Mexicans to do genetic and clinical epidemiological research in Mexico, not only in Tech, but also in Mexico. And I'm here and I'm very delighted to present Sir Rory Collins because one of the models of paradigms of biobanks is the UK Biobank. And as you will hear in the summary of Professor Collins' life, he has been and is Chief Executive of the UK Biobank. It is really my pleasure to present to this audience here at the Tech Science Summit of 2023, Sir Rory Collins. Rory, welcome and thank you for having with us this conference.
Sir Rory Collins. He is an epidemiologist who studies how to prevent and treat cardiovascular disease in large population-based studies. He trained in medicine at St. Thomas Hospital, London University, and statistics at George Washington and Oxford universities. He currently heads the Nuffield Department of Population Health. Since the early 1990s, he has been involved in conducting large-scale randomized trials of the effects of modifying blood levels of cholesterol. He became principal investigator of UK Biobank in 2005. This is the largest, deeply characterized prospective study of the disease globally, involving 500,000 participants. He was knighted for services to science in 2011. Elected to the Royal Society in 2015, and awarded the UK Medical Research Council's 2020 Millennial Medal for his national and international contributions to both cardiovascular disease and UK Biobank. Please welcome Sir Rory Collins. Sir Rory Collins. He is an epidemiologist who studies how to prevent and treat cardiovascular disease in large...
I hope that you can hear me. If someone could just confirm that you can hear me. Pablo or the organizers, can you just confirm that you can hear me speaking? Thank you very much. So first of all, thank you very much for the invitation to speak today. It's a great opportunity to talk about what we've been doing with the UK Biobank study, but more generally, talking about the need to develop large prospective cohorts in different parts of the world. And since the beginning of this century, not only have I been involved in the UK Biobank, but I've had the good fortune to collaborate with Pablo Curry and colleagues in Mexico on the Mexico City study. And I know that you're also planning other studies in Mexico City. So the points I want to make are, why does it make sense to establish very large prospective cohorts and follow people for a long period of time in order to understand the determinants of disease? Because these are very major undertakings. They involve substantial effort and investment in order to recruit large numbers of people. And they then take a long time following those individuals before they really start to be mature enough to allow studies of the determinants of disease. But the big advantage of prospective cohorts, that is studies where you record information about people many years before they develop disease, and then wait until they develop disease, is that you are able to measure the risk factor before the disease develops, and in particular before the disease itself can change levels of the risk factor. Avoiding, in particular, reverse causality. And we know that when people get particular conditions, that can lead to changes either in the way in which they live. They might stop smoking, they might change their diet, say with heart disease, or indeed the disease itself may change measures within the individual. And this is particularly important when we're looking at non-genetic risk factors, or when we're interested in the interaction between genetic and non-genetic risk factors, which I think is going to be increasingly important. The other advantage of having this large population study is that one can compare the people who develop disease with much more similar individuals who haven't developed disease than in the kind of standard retrospective case control study, where the cases may not be from the similar population as the controls, whereas with a prospective study, it's much more likely that they are similar in other respects. So confounding by other factors, that is where other factors are the determinants of disease rather than the risk factor of interest, is likely to be less extreme. And although they do take a long time, and although they do take a large amount of investment in time, effort, and money to establish these cohorts, in the long term, they are an extraordinarily cost-effective resource for studying many different determinants of many different diseases. So one can look at, say, a single exposure and its effects on many different diseases. Smoking was first studied in the British Doctors study to look to see whether smoking was causally related to lung cancer. But that prospective study then went on, as with people, the doctors were followed, to demonstrate that smoking was not only associated with lung cancer, but many other kinds of cancers, with other kind of lung disease, chronic obstructive lung disease, and vascular disease. So very efficient at determining the full effects, both bad and indeed potentially good, of a risk factor, or to look at the effects of many different exposures, genetic, biochemical, environmental, lifestyle, on a single disease. What are the combined effects? So when one establishes a resource like this, it can be used by many different kinds of scientists to study many different conditions in many different ways. But as I've indicated, it involves the establishment of very large studies. And the reason for that, of course, is that even in hundreds of thousands of people, only a small proportion of those individuals will develop any particular condition. And it's only the numbers of individuals who develop the condition that will determine the power to study associations between risk factors and the development of that disease compared with the people in the study that didn't develop the disease. So you need to have very large numbers in order to get enough cases of a particular condition. So what do I mean by large? Well, this is an analysis done by one of my colleagues, Sarah Lewington, who had been working for many years, bringing together a large number, about 60, observational studies, prospective cohorts that had been established in different parts of the world, looking at the association between blood pressure and the risk of cardiovascular disease. And in total, she was able to pull together, collaborating with researchers around the world, a total of about 1 million individuals that have been studied for over 10 years in terms of follow-up, looking at blood pressure on cardiovascular disease. And so I said to her, well, what would have happened if instead of having 1 million individuals, you only had 5,000 of them? So she randomly selected 5,000 to look at the association of a major risk factor, blood pressure, on a common condition, coronary artery disease mortality here, and the interaction with another powerful risk factor, in this case, age. And this is the graph that you're seeing now. This is the association between the risk of death from coronary artery disease at different levels of blood pressure and at different ages. And the vertical axis is a doubling scale. So we're looking at kind of exponential slopes. And when you look at this 5,000 people in this study, it's very difficult to determine what the associations really are between blood pressure and the risk of coronary artery disease mortality. There's a general upward trend, but the confidence intervals, these vertical lines through the dots, which are the point estimates, are very wide. So you can't really get a sense of discrimination between the risk at different ages and different blood pressures and the shape of the associations. So then I said, well, what happens if you have 50,000 of these people selected at random? So things are starting to come into focus. But when we look at this, is there a flattening at the older ages whereby higher blood pressure is not associated with higher risk? These were the kinds of data people were looking at in the hypertension field and saying, well, high blood pressure is actually not a risk factor in the elderly. There's no benefit in lowering their risk because the curves flatten off. In the younger individuals, is there a J-shaped curve whereby lower blood pressure is associated with increased risk, so that you could cause harm by lowering their blood pressure too much. And then we said, well, what about 500,000 people? And now everything comes into focus. It's like having a microscope, and suddenly you can see the structure with a microscope of the cells. With this metascope, if you like, you can see the structure of the association between blood pressure, age, and the risk of coronary artery disease death. And we see these beautiful log linear associations. So the confidence intervals, those vertical lines, have become much more narrow. We can see that they are straight line associations at different ages. So they're log linear associations, which means that each difference in blood pressure is associated with the same proportional difference in risk within any of these age ranges. In addition, you can see the slopes are different. So the slope is not as steep at older ages than at younger ages. So in relative terms, because it's a log linear curve, in relative terms, the same absolute difference in blood pressure is associated with a bigger proportional difference in risk at younger ages. But because it's a log linear association, a smaller relative risk at older ages is actually associated with a bigger absolute difference in risk for any given blood pressure difference. So you can see how important it is, even with two strong risk factors, age and blood pressure, even with a common condition, to have at least 500,000 people to be able to understand these interactions. And when we're wanting to look at more subtle associations for less common conditions, then we need to be thinking in terms of having millions of individuals in these large prospective cohorts. And for reasons I'll describe in a minute, why there would be value in having these in different populations. And the reason for that is diversity. We want to have large numbers of people in different environments. We want to have we want to have people who have a different, both internal, that is genetic and ethnic environment or risk factors, exposures, and different external exposures, lifestyle, environment, the social determinants of health, for example. So having a single big study in one place will not give you that diversity. So when we think about this, why is it important to have a strategy of establishing some large, very well characterized cohorts with long-term follow-up? Genetic heterogeneity between populations may well be important in understanding the determinants of disease, because different genetic variants are more or less common in different populations. If we have different populations then we will get a wider range of exposures. So back before the commercialization, the capitalization of China, there was very little access to meat, dairy, and people were very, very thin, particularly in the rural areas. In Mexico, the diet is associated with more obesity than we see in China or in the UK. So if we want to study the full range of exposures in humans, populations, studies set up in populations with different levels of adiposity or different levels of blood cholesterol because of different diets or other kinds of exposures will increase our ability to study the full effects of those exposures. An example might be if we were interested in smoking. If we only did a study in a population where everybody smoked either 10 cigarettes a day or 20 cigarettes a day or 30 cigarettes a day, and we compared risk between the people who smoked 30 cigarettes a day and those who smoked 10 cigarettes a day, we would underestimate the importance of smoking because we would not be comparing it with normality, which is 0 cigarettes a day. And the same is true with other kind of risk factors. So here's an example from the Mexico City prospective study that we've had the pleasure of collaborating with the investigators in Mexico on. And these are taken from a paper that they published in the New England Journal on obesity and diabetes. So here is in that study in 2 parts of Mexico City, at the baseline near the beginning of the century, the body mass index level of obesity in women and men. This then shows the rates of diabetes in women and men at baseline in this study. And you can see that at age 60, something of the order of 20–25% men and women had diabetes at entry to the study when they were age 60, compared with rates in the US of 9% or the UK 6%. And largely it will appear driven by high body mass index. In addition, in the people who had diabetes, their HbA1c levels on average were much higher than the levels in people with diabetes in countries like the US or the UK, 9% versus 7.2%. So higher rates of diabetes, but also among the people with diabetes, higher levels of HbA1c, indicative of not such intense control of their diabetes. And then when we looked at how that translated into mortality, we're able to now look at, among the people in this study, the effects of diabetes on different causes of disease. So this goes back to the value of this, this kind of prospective resource for studying the impact of a single risk factor on many different diseases, and also to get a quantitative estimate of the relative impact on different conditions. So for renal disease here, we see diabetes is associated at younger ages with a 30-fold increase in the risk of death from renal causes. In 60 to 74-year-olds, a 14%, and even in the elderly over 75, a 5-fold increase in the risk. And then for cardiac diseases, we're seeing somewhere between 2- and 5-fold increases in the risk of cardiac mortality, depending on age, which is about twice as much as has been seen in the US or UK studies. So we're able to study the impact of that condition and try to understand why the relative risk might be so intense, and then think about what we could do from a public health perspective in terms of ameliorating those effects. So increasing the diabetes control in people with diabetes. Diagnosis was very high. So when we looked at HbA1cs in people who did not report they'd been diagnosed, very few had undiagnosed diabetes based on HbA1c. So diagnosis was occurring, but the control of the diabetes was not as effective as it could be. Moreover, those individuals were not getting other cardioprotective treatments, which are like blood pressure control or blood cholesterol control, all of which would reduce their risk of cardiac and indeed other complications. The other reason why one would think about having studies in different populations is that some diseases are rare in one population and common in another. And so if you want to study a particular condition, it may well be worthwhile going to a population where that condition is common. So if we look at the mortality in different parts of the world, and I'm going to focus here on stroke, in the US, for men aged 35 to 69, the age-adjusted mortality rates from stroke, about half those in Japan, but about 1/8 of the rates in urban and rural China. And indeed, when one looks in the UK and the US are not so dissimilar. Because stroke is so common in China, actually, even 10 or 15 years ago, the diagnosis of the type of stroke using imaging was far better than in the UK, because it was a common condition, and therefore the healthcare system was set up to try to diagnose what kind of stroke and to do imaging very early on, which then makes it much easier not only to have larger numbers of strokes, but to have better characterization of what kinds of strokes those individuals have had in this population. And here are some data from the China Kadoorie Biobank of 500,000 people set up by the Chinese Academy of Medical Sciences, working with my colleague Chen Zhengming, looking at the association between systolic blood pressure and body mass index, showing a very strong association of body mass index with systolic blood pressure, so a major driver of increased systolic blood pressure. And then the middle and the right-hand graph showing the association between systolic blood pressure and ischemic stroke and hemorrhagic stroke. And what you can see is that a 10 millimeter difference in systolic blood pressure is associated with about a relative risk of 1.3, a 30% relative increase. Whereas for hemorrhagic stroke, the relative risk is twice as great. The slope is twice as steep, again on a doubling or logarithmic scale. So the relative risk is 1.6, a 60% increase per 10 millimeter difference in systolic blood pressure. So again, having large numbers, you can see the point estimates have very, very narrow confidence intervals. There's those squares, the confidence intervals are within the square, they're so narrow. You have a very beautiful log linear association again. So we have very clear picture of the shape of the relationship. And we can discriminate between the strength of that relationship for ischemic and hemorrhagic stroke in this population. And then, of course, the advantage of having a series of studies in different populations is that a finding in one study can be tested in another study to see whether it's reinforced. And of course, by combining studies, one will get increased power to study, not only to confirm an association, but to investigate that association in greater detail, to look to see whether there are interactions that can be studied when you have much larger numbers by combining different studies. And here is an example from some genetic work involving UK Biobank, UKB here, the Geisinger Health System, a HMO based in the US, and the Mexico City prospective study, the one I mentioned before that Pablo Correa and his colleagues are running in Mexico City and was set up with the Mexican Ministry of Health. This was a finding of a novel genetic association through exome sequencing that found a loss of function variant within the gene GPR 75 that was observed in the UK Biobank study to be associated with a substantial difference in body mass index. So a potential new target for treatment of obesity. The Geisinger study reinforced that, and the Mexico City study reinforced it further. And the UK Biobank study generated the hypothesis, so you'd want to know that that's confirmed by other studies, and here it is, of course. And then when you combine all of these studies, the meta-analysis result has much narrower confidence intervals, so it allows greater exploration of the association in different settings, and to see whether this association on body mass index is associated with certain conditions. I should also mention that the Mexico City study, although smaller than UK Biobank, contributed about the same amount of information because body mass index is more common, high body mass index is more common, but also because the genetic variant was more common than in the UK Biobank study. So again, illustrating the advantage of having genetic heterogeneity as well as complementarity between the different studies. So it's heterogeneity that's really important, to have different population studies in different settings with different exposures, but internal and external exposures, different rates of disease, so that one can look at the full effects of many different exposures on many different diseases. So I've talked about size and diversity, but I think it's now increasingly important to get detailed characterization of the individuals in the cohorts that are set up. So there's an interesting balance between lots of very large studies that are very shallow and a strategic set of studies in different settings, in different circumstances, with, as I say, diversity of exposures, where you have deep information about those individuals and their different exposures. So first of all, you want to get a lot of information from the participants, detailed questions about their lifestyle, their medical history, their exposures that you can get from asking them questions, by measuring them to get at things like their body mass index, their body impedance, their blood pressure, their lung function, etc. If one can go deeper than that, so for example, in UK Biobank, with 500,000 people, we're currently in the process of imaging with magnetic resonance 100,000 of those individuals to get very, very deep information about them. So magnetic resonance imaging of the brain, the heart, the body, DEXA of the body to look at body composition as well, and bones and joints, carotid ultrasound to look at atherosclerosis. So very, very deep. But remote monitoring, so getting people to wear activity monitors. You can ask questions about activity, but they're not terribly accurate. But you can put activity monitors onto people, ask them to wear them for a week, mail them back to you, download the data, send out the activities monitor to another person. So they can be recycled, and it can be a very cost-effective way of getting information, accurate information about people's exposures in terms of activity, or it could be exposures in the home. In Britain, the second most common cause of lung cancer after smoking is radon. That seeps out of the ground and is very variable house to house. So you can put monitors in the home. So getting deeper information about people's exposures. Also, we know that exposures change. So going back to people and finding out whether their lifestyles have changed. Have they stopped smoking? Have they changed their diet? Have they changed their activity? So repeated assessments in terms of questions and measurements, but also in terms of biological samples. One would ideally collect as much biological sample, in particular blood. Blood is particularly valuable to look at genetic, but also other omics, proteomics, biochemistry, hematology, and the ability to look at these things at scale is becoming increasingly possible. So we've seen in the last 20 years, moving from sequencing the first person to, in UK Biobank, we've now sequenced all 500,000 participants in UK Biobank. Not just genotyping, which we've done, not just exome sequencing, which we've done, but whole genome sequencing of all of the participants. And following on from that, we're now seeing approaches that can be done at scale with omics. So Nightingale Health, a spin out from a Finnish university, has developed an NMR method that uses very, very small amounts of blood to measure lipidomic and lipid-related measures in hundreds of analytes and fatty acids. Companies like Olink, SomaLogic, and some of the mass spec approaches are getting to the point where they could, within the next couple of years, do proteomics and metabolomics on 500,000 people and produce data on thousands and thousands of proteins to combine with the millions of genetic or billions of genetic measures one would have on participants. And so I think we're seeing the technology catch up with the cohorts that were established 20 years ago. The Mexico City study, which was a work of genius by the Mexican Ministry of Health and the team that developed that. Because 20 years ago, no one was thinking that this was the way to go. The technology is now caught up with studies like that. And so studies that collect biological sample, blood, urine, ideally stool to look at the microbiome, and to ideally collect them in different types of tube in order to be able to look at different analytes. Having said that, though, I think the technology is getting to the point where it can measure many, many different things in simple blood samples. And then ideally, wait until those assays can be done on the whole cohort in order to generate information that will be relevant to many different diseases. So you thinking about creating a resource. And this is one example that came out of that kind of approach. So in UK Biobank, which started recruiting participants in 2006, recruited 500,000 people across the UK, United Kingdom, aged 40 to 69 at that time, men and women. In about 2015-16, funding was made available to genotype the whole cohort. So using an Affymetrix array, which was designed for UK Biobank, measuring about 800,000 markers across the genome. As you know, there are about 3 billion base pairs across the genome. So measuring, you're only around 1 million. You're doing them at quite wide intervals across the whole genome in an individual. But you can combine data from genotyping with sequence data known for a particular population in order to estimate, or to use a technical phrase, impute the variants that you haven't measured that are near to or in the region of the variant you have measured with your genotyping. So you can estimate with decreasing accuracy, the further away you get from the genotype you've measured, what the genotypes are that you haven't measured. And you can then combine these data to generate so-called polygenic risk scores. And this was something that was done in UK Biobank and done for the first time, actually by a PhD student at a university in America, because the data were there. So what one's doing with UK Biobank or with the Mexico City study is generating resources that allow researchers to do research that otherwise would not be possible. And in this case, this was research done by a PhD student for his PhD, for his doctoral thesis, that would never have been possible without building that resource. Now, before these data became available, it was thought that these genotypes, which were likely to have only very small effects on disease, they might have a small increase or a small decrease in disease. And therefore, it was hypothesized that they couldn't be useful for predicting disease, because people would have a random set of different variants. Some would increase their risk, some would decrease their risk of that particular disease. And so you wouldn't be able to identify people who are at high risk using these variants. The data, however, contradicted that common view. And these data showed that actually, if you combined variants of small effect, and this PhD student did it for coronary artery disease, then you can identify about 5% of the population for coronary heart disease that have a risk equivalent to someone with familial hypercholesterolemia, which in the UK is about 0.5%. So you have a 10 times higher yield of people who have equivalent risk of coronary artery disease to someone with a single gene disorder. And then this work has been extended by other groups to show the same is true for, say, breast cancer. You can find about 5% of women with a polygenic risk score that puts them at risk that's equivalent to having BRCA1 or BRCA2, a single gene disorder. Or prostate cancer here, or type 2 diabetes. And indeed, Peter Donnelly in Oxford demonstrated that with a single genotype, where you do 800,000 markers, you can generate a series of polygenic risk scores for different conditions, such that for, say, 16 conditions, about a quarter of the population will be in the top 5% for at least one of those 16 conditions. So you think about this. Genotyping is now relatively inexpensive. It can be used, a single genotype array in an individual can be used to identify people who are at high risk of many different conditions. That could be applied in, say, screening. So in Britain, where we have breast cancer screening, which is largely driven by age, it could determine a much more refined age-based screening. So you might take the 5% in the top polygenic risk score and screen them at much younger ages. Those in the lowest 5%, you might never screen them for breast cancer. For prostate cancer, when men get to a certain age and they have a PSA done, then it's very difficult to know what to do next, because it's not a very good predictor of risk of prostate cancer. You can get false positives. But if you were in the high 5% for prostate cancer on a polygenic risk score, and you had a high PSA, your posterior probability, combining those two together, would indicate that maybe you need to be investigated more. Or when do we use statins? Again, largely driven by age and gender at the moment, but you could identify, rather than people who have familial hypercholesterolemia, the 0.5% in the population, the 5% in high polygenic risk score, and think about giving them cholesterol-lowering therapy or offering it to them in their 30s and so on. So I've talked about size, I've talked about diversity, the need to have large studies in different settings. I've talked about depth, about the importance of getting detailed information about the exposures in the individuals. And then finally, I want to talk about follow-up. So the key thing is to establish these cohorts in settings not only where there is diversity, but also where it's possible to follow up their health efficiently, ideally in detail, ideally in depth, as with the example of stroke in China. So when we talk about depth, I talked about detailed questions and measurements, deeper phenotyping of the participants and their exposures, the collection of biological samples, but also deeper phenotyping of the diseases that people develop. So as I said, with stroke in China, the imaging data on the strokes, allowing the separation of ischemic and hemorrhagic strokes, is very important, because it showed that blood pressure was twice as important for hemorrhagic stroke as for ischemic stroke. And having more detailed, more specific information about the diseases is important in two ways. So it enhances the power to detect associations between risk factors and disease, And there, the main problem is you think someone's got a condition and they haven't. So false positives will weaken the association. The second one is increased specificity. So my example of being able to separate hemorrhagic and ischemic strokes, because a risk factor may have different associations with the different subtypes. And indeed, it may have an association with one subtype and no association with another subtype. And here's an example of that. So if we look at reproductive factors such as parity, age at menarche, age at first birth, then these reproductive factors are largely, if not wholly, associated with breast cancer with positive receptors. So ER and PR, estrogen and progesterone receptor positive tumors. And they're not really associated with the negative tumors. So we're seeing a different biological process going on, demonstrates the importance of being able to separate these different types of breast cancer. And when we think about the follow-up, the ideal would be to collect the outcome data in the same way that we collect the biological sample, not knowing necessarily what we're going to do with it, because it may be possible to go back to the information we have on a disease and apply new knowledge to be able to subtype the disease that we couldn't do at the time of diagnosis, provided we have information. So let's say tumors. If someone has developed cancer, if one could get a tumor sample and store it, then as greater knowledge becomes available, maybe one would go back to those tumors and be able to analyze them in greater detail. So in UK Biobank, we're now looking at digitizing the histopathology slides for individuals who've been diagnosed with cancer. And then various groups are applying AI and machine learning to that digitized histopathology data to pull in much more information out of it than was pulled out when the diagnosis was being made by a pathologist. So in conclusion, the strategic approach that I think is really important to think about is establishing a number of large cohorts in different places that allow us to look at and investigate genetic and other diversity, that allow us to generate genetic and other assay data, that collect other types of data that characterize the exposures in that individual. And finally, to remove obstacles to data access and sharing. With UK Biobank, the Wellcome Trust, the big charity funder in the UK, and the UK Medical Research Council, the major government funders that set up UK Biobank, required it to be set up in such a way that the data were available to researchers anywhere in the world to use on exactly the same basis. So it's as accessible to researchers in Mexico as in the UK. It's accessible to researchers in industry as in academia. And the reason for that is that having built this resource, what success means is as many people as possible using the resource to make novel findings, to do discovery science. And different people will approach the same data in different ways and learn different things from it, much more than any single research group could do. So in UK Biobank, what we have been doing is just saying to researchers around the world, the data are available. I think some people find it difficult to believe that. And we see use of UK Biobank in some areas of the world and not in others. So we see high use in the UK, in the US. We went to China in 2019 and had a meeting with the Chinese Academy of Medical Sciences and said the data are available to use. And over the last 3 years, we've seen China become one of the biggest users of the UK Biobank resource to understand disease that will be relevant to the Chinese population, even though The study is in the UK. We send the data to researchers for them to put on their own computers. As the data have become much more complex and much bigger scale in UK Biobank, we've now built a cloud-based research analysis platform so that rather than the data going to the researcher, where the researcher would need to have their own computers and the facility to analyze the data, they can actually go to this analysis platform that's based on the Amazon cloud, based in the UK, and they can go to the data. They can use the tools that are on the platform. They can bring their own tools onto the platform, and they can do their analysis there. And in order to remove obstacles further, Amazon have made available $500 million of free compute on that platform to all early career researchers anywhere in the world, but to all researchers in low and middle income countries. So throughout Central America, South America, Africa, India, any researcher who wants to use the UK Biobank data can get access to these data, and the computing power to analyze these data effectively for free. And so in the same way that I imagine researchers in Mexico will start using increasingly the Mexico City study, I hope that this talk will provide an invitation to you to come and use the UK Biobank data as well. And in fact, we have been collaborating with the Mexico City study to take what we've learned in terms of building a platform that's cloud-based to do the same thing for the Mexico City study so that it's readily available for researchers as well. So my final comment is we don't need lots of large cohorts. We need to have a number of informative cohorts in carefully selected settings. I think the China Kadoorie Biobank is interestingly different from the UK Biobank study. I think the Mexico City prospective study is interestingly different from the UK Biobank and the China Kadoorie Biobank. And that they are probably among the three both biggest and longest, the most mature studies that exist. Certainly there are new studies being set up now, but they will take 10 years or so before they mature. So I think looking at what exists now, thinking about how the data can be made even more valuable by assaying the samples, and the Mexico City study now has exome sequence data and genotype data on the whole cohort. It will soon have NMR lipidomic data on the whole cohort, characterizing the health outcomes in that study as well in greater detail, will put Mexico in a fantastic position, but establishing new studies in places like Mexico as well as Africa, provided one can follow up the health, will be an important step forward in understanding the determinants of disease and understanding ways in which to treat and prevent disease. And I go back to my example of the Mexico City study to find major determinants of disease, BMI on diabetes, causing very large numbers of deaths from renal disease, that led to public health measures that make changes very rapidly. So this is not about publications in journals. This is about really improving the health of our population. So thank you very much for the opportunity to speak. I look forward to many of you applying to access the UK Biobank data, and we will do everything we can to help you to use it, and look forward to continued collaborations with our friends and colleagues in Mexico. Thank you.
I just want to finish, Rory. Thank you for your time, for the preparation, and for the message that you have conveyed to us about the importance of setting up large prospective studies. I just like to also mention that you have been heard for more than 300 people here in presence and that have been connected remotely to the conference. It's really a pleasure for Tec de Monterrey to have the opportunity to hear you. And thank you to the audience, and Rory, really thanks. We don't have, unfortunately, time for questions and answers. We have to continue with the program, but we'll be in touch for sure with you and continue learning from the experience of the UK Biobank and the research that you've been doing in the area of chronic and non-communicable diseases. Thank you again, Rory. We'll be in touch. Thank you.
Thank you very much.